O301 should be on.
Sorry, this is O301 and O302.
Are combined.
Yes.
Perfect, so they're already on.
Yes.
Yes, okay.
Thank you.
Yay. Thank you for asking. And also, now that you have, helped me figure it out. Anytime that doesn't happen, just email me, because it should happen. As soon as I lecture, it should go on at least be on that morning, but it should be on soon.
Okay.
You shouldn't have to wait at all.
Okay.
Okay, cool. You ready? Let's work on it for a minute. We'll see how far we get. We're going to do another biomolecule. So far, what biomolecules do we have on the exam? Lipids, right? So we already reviewed that. We're doing that. Now we're going to do carbohydrates. Okay? Large class of naturally occurring polyhydroxyaldehydes and ketones. Again, this is nice because it has kind of one primary function, and that's immediate energy. So, a secondary function, though, is backbone, is structure, which is very unusual. Now, it ends up in the backbone of our DNA. It's the structure that makes the DNA. It makes the double strand. It's part of that. And then the other one is plant cell walls are literally made out of carbohydrates. But I'll tell you ahead of time, I can't digest them. So when I eat a plant cell wall, give me a celery stick. Right? Or, hey, here's a carrot. That cell wall just pushes on through. And then we call it fiber. And it's like a plunger to clean us out. I'm sorry, but it's just the truth, right? And so that's what we say when we need more fiber in our diet, because plant cell walls we can't digest. So they go through our digestive system and go, oh, I'm gone. And it cleans out everything in our intestines, which is kind of nice. That's a good function for it. A lot of animals, though, like cows, right, they can eat grass and they literally can start digesting that and turn it to sugar. But out of the vegetables and the plants we eat, we can extract other things that are in the cells. So the cells get, mechanically broken apart, and then we pull stuff out of their vitamins and minerals. Does that make sense? So, I'm just kind of giving you a heads up on that. So, let's do what we usually do, what I'm going to kind of try and do in the biochemistry. I want you to be able to identify pieces of it. So we're going to start with kind of simple sugars. They sometimes get this word monosaccharide. So we're going to do it in 3 groups. We're going to do the simplest of sugars, which we call monosaccharides. We're going to do two sugar. In other words, smack a couple monos together and you're going to have a disaccharide, because that's very prevalent in our diet. And then we're going to do polysaccharides, which now that you understand polymerization, if I took a monosaccharide, see how they're kind of using the wording like a monomer, and it started to polymerize, it'll make this polysaccharide like this big old complex sugar that's a polymer. All right, now, just some words. We're going to talk about aldoses and ketoses, but I'll show them in a second, but I'm going to just get you this idea down. If in the sugar, and I'm drawing all this, this is the structure, this is the hydrocarbon chain where really the thing that identifies the sugar is hydrogen. I kind of hidden from you for a minute. There's one piece that's an aldehyde, so I called it an aldose. One piece called do you see how this is a ketone? So that's called the ketose. So in the naming, you might hear about aldoses and ketoses. Now you're going, oh, those other two are different. But when we do sugars, it's very simplified. You just go find the carbonyl group, aldehyde or ketone, move away from it to the last chiral carbon, and then figure out if it's left or right. Okay? That all making sense? This is the actual thing your body takes in, it's glucose. But if you look at it, it's very similar to this, just a couple little carbons twisted up here. But the one that your body naturally uses is the D-glucose. It wouldn't know what to do with L-glucose. And what they've done in sugar science is they basically say, if these are close enough that they'll trick my taste buds, but they won't trick my body, then sometimes they make them into artificial sweeteners. And that's kind of your business. That's what you say, hey, it's okay to eat that for your teeth. Maybe not that great for your liver, but it's good for your teeth, right? You with me? Because your body can't turn it into any, and also bacteria can't either. They don't know what to do with it either. They're also wired for these sugars on this planet. Make sense? So, that's kind of a side note, like how they make these artificial sweeteners. One way. I'll show you another way. All right? All right. So here we go. First thing, put priority group on the top. You got to get that twisted up sometimes. So, here's a fun one to play with. We'll go back to this one. I have one laying sideways. He's laying sideways, so I don't know whether that... So, okay. So, here we go. We're going here, and I'm going to just number this up so you guys can talk to me for a second. So where's the farthest chiral carbon from the carbonyl group? That's chiral. 5. Okay, good. And I'd love to say it's right or left, but it's not until I twist that up.
Okay.
When I twist it up, which way is this thing going?
Right.
Right, so this is a D sugar. Cool. So you have to practice that. The nice thing is when you're taking your test, I'm okay with that. You guys are on camera and all that stuff. That's okay. And I'm sure your lockdown browser is going to go, there's something weird going on. You can twist your old computer if it helps you. Like just go and see, get the thing up top and see which way the bond goes. I'm cool with that.
Okay.
If I see students taking the test and they start twisting their paper, I'm like, yay for you. I mean, why risk like in your head twisting it and going, oh, I got that wrong. Just twist it. Put that carbonyl on top, and then that will naturally go to the right way. I can't quite do that here because I'd have to jump up there and go crazy. Is that good? So that's fun. There you go. There's another little cool, very organized piece of nature. I just think that's so fascinating. All these sugars in there, it's like, no, the only thing that my body knows is D sugars. It's really wild. And again, you can imagine there's a longer story here because we're not even messing with all these, flipping them right, left, and all that. But just so you know, that's what the difference between, like, you hear lots of single sugars, like, okay, that's glucose, that's fructose, that's ribose, that sort of thing is what they're doing. They're just some of those other things twist, and again, there's a whole field of carbohydrate chemists. All right, so we kind of got that going. Everybody feel good about it? And you also kind of know what the Fisher projection is, why we're just drawing them with straight lines. So horizontal lines are going which way? Out towards you. So if you're looking on your paper, they're coming out at your face. Then the vertical lines, they're going back into the paper. And then if you sometimes see this going, like sometimes people like to be very specific, just so you know, triangles are coming towards, and then the dots are going behind. And usually with dots, depending on what the program is, it's writing it. Sometimes they just get drawn out like that. That means it's going behind the board. Follow? Is everybody okay with that? Does it make sense what I'm kind of saying?
Where would they put that?
Yeah, so here, let's just say I'm going to do something like this. I'm going to make something chiral, I'll put something different on each group. Does that sound good? I want to show that these are coming out at you, so I'm going to actually do that. And then I want to show these are coming back, so I'm just going to, like that. As opposed to, I very seldom see people thought it like that. And I very seldom see people that are so, sloppy. But it looks cool. It looks neater when other people do it. All right, so there's that. Kind of fun stuff. Okay, here, we'll get to try some. So I want you guys to try these. I got 1, 2, 3, 4. Now you can see what I've done here, right? Can you see what I've done here? Yeah, I gotta do a big flip on this thing. So try that. See if you can get all 4 of these and decide if they're D or L. That's all I've had. And while you're at it, will you then? So I gave you the name, right? But could you give it the official name? I mean, this part you don't know. You don't know ribose from mannose from aldose. None of that means something to you. But you could do D and L, right?
Mm-hmm.
But while we're at it, you might as well practice the stuff we do know how to name this, right? Aldehyde or ketone? How many carbons?
5.
5. So what would you call that? That's an aldopentose.
Pentane.
Aldopentose.
Dose.
It's an aldopentose. Is it that one D or L? Okay. Okay. See if you can do these other ones. I'll give you a minute. It's fun to think about. If you need to just take a picture, but it's on your PowerPoint slide, so you can download that too. Have you guys seen these rocket books, by the way? It's my new favorite toy. They're reusable notepads. You basically just get there's like a cloth. You just wet down one corner and erase everything when you're done. But the minute you're done, you take a picture of this and it sends it up to your Google Drive or wherever you want to send it.
Oh, that's cool.
Yeah, so it's very cool. But the best part of it is that, and I'm getting them, I haven't tried them yet, but you can get magnetic borders for your whiteboard, and if you guys all have that app, which is free, the Rocket Book app, you can actually screenshot the board and it goes right up to your Google Drive or your email, wherever you want to send it. So I can send it to different things by how I mark right here. I got 1, 2, 3, 4, 5, 6, 7 different choices of where I want to send it. So when I mark it, take a picture. So when I'm working with students tutoring, right, I'm always writing, we're talking, writing, then they're like, can I have that? And I'm just going to be like, goes to your email.
That's smart.
It's pretty smart.
Yeah.
These are like $20. I'm not selling them, I wish it was, but the whiteboard things are ridiculous because they're $12. Those smart boards, the electronic, those are like $2,000, $2,500. So I'm like, I'll spend $12 and just take a picture of the board. Oh, and also it transcribes it, so it's not like your picture, right? You're like, well, then why wouldn't I just take a picture, right? You know how your pictures, it's kind of hard to blow them up? This actually transcribes it up to an 8.5 by 11 image automatically, and it darkens in lines. So it's pretty cool. Yeah. And these pens are erasable, so that's kind of cool. These are just normal erasable pens.
That's really cool.
I'm kind of excited about that. Everybody I know that teaches high school, because I used to do that way back. But so the guys I start teaching with, they're girls. They're all principals and stuff now, so I send it off to them because a lot of them are in districts. They can't afford a smart board. So they're like, dude, $12. Everybody in that district has a smart board. Perfect. Rocket Book. Okay. We ready? I think we nailed the first one. So number 2, what do you think about number 2? Somebody over here, D or L? D, because it's on the right-hand side or on the left? Okay, so here we go. We're going to start at the top. Where's the, if that's 1, that's 2, that's 3, that's 4, that's 5, where's the chirocarbon? Oh, okay, so it's on the left, huh? So then what would you call that? L. It's an L sugar. Bonnie wouldn't know what to do with that. If we're going to be a little more specific, though, aldo or keto this form?
Aldo?
Aldo, and then 1, 2, 3, 4, 5, 6. That's an aldohexose, L form, right? Whoa. Uh-oh. You guys ready? Go to this back core over here. When you twist it, I'm going to twist it up, right? Which way is that going to be then? D or L? This one. Which way is that going to end up when I twist this thing up? D, it's on the right. You guys see it? Here's plan B. I saw you doing it. Right? I'm like, okay, I'll just do it this way. Yep, that's on the right. That's right. That's fair too.
I didn't even catch myself doing it.
I know, but it's very good. That'll work. That way you're just perfectly certain. That's the importance of that, right? 1, 2, 3, 4, 5, 6, right? Although hexose again, but it may be different in these. You'd have to check all the sugars. Maybe the same thing. I don't know. Ooh, here comes one. This guy's just got to go a whole 90. Now this one's going to be hard if you want to do it with your head, right? You can stand on your head. I used to stand on my head all the time when I was a kid. I don't know. Walk on your hands. Once this gets flipped up, what do you guys think? Down on this end, is it going to be D or L once it gets flipped up?
D.
Goes all the way around, and that thing's going to go to this side, right? So that's a D sugar too. Okay. Ooh, that's an interesting one. What is that? There's a carbonyl group, there's a carbon, there's a carbon. What's that called? Yeah, so this is a ketose. How many carbons? 6 again. That's a keto hexose D form. Perfect. That's it. Oh, I flipped it. Nice. Nice. Now, this is interesting. This has caused a lot of problems in the food industry and whatnot because it's really hard. Like all the instruments I have that can detect, they could figure out that there's 6 carbons. They could figure out there was alcohol groups. I could figure out the molar mass. Do you understand that the molar mass? Remember how we did that? Count carbons, hydrogens. You'd figure out the molar mass of this thing. I couldn't tell a D from an L. I have no tool to do it. But it caused problems because, for example, I don't know if you remember, there was an incident where we were getting dog food that was actually, the protein was produced in China and sent over, and it was the same thing, that DL thing. And they were sending L form. And so not only could the animals not produce it, it actually poisoned them. So they couldn't. So it was really crazy. So it was like, wow, we need an instrument that'll kind of figure this out. So, this is kind of wild. When polarized light is passed separately through this fancy word, enantiomers, that's another word for the chiral carbons, they rotate the light. Now, when we call them enantiomers, what we mean is the D and L are enantiomers. Like me and my brother born on the same day, we're twins, right? The D and L, right? Me and my brother, same day, same womb. Okay. We're twins, but the D and L sugars, they're called enantiomers of each other, if everything else was the same. So it's interesting, though, that what happens when polarized light passes through a column of this sugar, like in solution, it'll rotate the light. And the wild thing is D rotates it one way and L rotates it the other way. So I think that's super cool, but I was thinking, like, if I told that to you guys, a lot of you would be having a lot of questions, like, what's polarized light, right? Is that anybody in here, like, it sounds cool, but, and then, right, and how do I know it's rotating? So I just got crazy, and I thought, I think I can show this to you. We'll find out. You guys ready? Come over here. This is kind of cool. You got to get up there just because I'm up. Come over and have. Check this out. It's kind of cool. So if you fish, you probably realize this, that when light comes out of that light bulb like this, it's vibrating. it vibrates like this and then moves like this, right? It's called a transverse wave. The light wave kind of vibrates, and then and off it, like it's heading towards you, it's doing like this. And if it vibrates faster, it's changing energy. That's changing colors. So purple's like this and red's like this. And sometimes I do that with my voice because my voice is the same thing. When I go high frequency, or I can go low frequency, right? That's the same idea. You with me? So, but light doesn't just go up and down in one direction because the light doesn't understand gravity and up and down. It doesn't know any of that. So when it leaves, it could be bouncing any direction. The vibration could be going in any direction, but the propagation, the way the light's moving, is towards you, certainly, right? You're like, oh, I'm light night, right? Is that kind of making sense to you? Okay, so we came up with these chemicals that are pretty cool because what they do, they have these big chemicals get excited at certain colors. That's why we get colors, right? That's what's going on, right? The chemicals absorb everything except green, so green comes out towards you. So what we've done is we've got these chemicals in here, and they very carefully aligned them so that when the light comes in here, it just cuts down to one vibration. And so it looks like it cut out a lot of light between us, didn't it? Mm-hmm. And to prove that they cut out one vibration, I very carefully marked these so I know what's going on. You see that, right? Make sense? If it cuts out only one vibration, it's now vibrating like this. Keep your eyes on that and watch when you do that. Do? So keep your eyes on my eye. Do you see what's going on? Are you not seeing it? You see me? Not very clearly, but you see me through the two pieces of plastic. Keep an eye on me. Yeah, that's what happened was if that vibration's coming through both, I just cut out that vibration with that second filter. And that's what polarizing filters are. Yeah. So you see me? Isn't that wild? So, and that's the variations we have, like your glasses do it, right? They get in the sun and they have a magical, I don't totally understand exactly, but it's working on the similar principle. It's working on, based on light, it does some sort of adjustment like that to cut out light, right? Does that make sense? So, you know, if you're a fisherman, it's kind of wild because when light goes off a surface like that, it does that automatically. So if the light comes in like this, when it bounces off, it comes like that. That's the only orientation that's there, which is great because when I'm fishing, I just need glasses like this because the reflection is totally taken out. And the only thing I see is the light that literally is coming off of the fish out at me, still kind of coming out at me like this. And that, so I can see through the water better. And that's why you guys get polarized light when you're on the water or skiing. Same principle. Light comes off a flat surface, it comes to one vibration, which is pretty wild. But just so you know, this trick I'm showing you will save you a lot of money. Because if you go shopping for polarized glasses to take skiing or fishing, and they're trying to sell you all this, you know, go to the Target cheap bin and put the glasses up by each other and twist them, and they'll go dark, and you're like, yeah, they're polarized. I'll spend $10. That's fine. Does that make sense? You know, a lot of people like overprice them. They're not that expensive with technology. All right, all that was to say, when light goes through a sugar, it twists like that. Like itself, the light itself gets twisted. So I could prove that if I could polarize the light to begin with, so I only have one wave going through the water, right? So I'm thinking this might work. No promises, I haven't done this before in this manner. But what my thought was, so you can kind of see the experiment. See how I'm doing it? I just got it lined up on the bottom of the beaker like that, and then I'll just pass light through. And then I'm just going to get normal water, like this. There we go, see what kind of mess we can make. And you know, it's always fun when you mix water and electronics. It's always a high, you know, things are going to go well. you go like, yeah, that's kind of being in Dr. Thompson's class. There's something crazy about that. All right, so at this point, nothing, right? Because the light is just coming through a polarized filter. It's not reflecting. I'm getting pretty much all of it. And then it takes me a minute to kind of get it centered on the ball, but this is it. All right, so I'm going to do it like that. All right, now, if this goes well, come over here, underneath there. There it is. Now it's kind of centered. So all I'm doing is focusing on the center of right there. That's what I'm doing. So you could guess, still getting light. I should be able to turn it 90. If I stay 90, should knock out that beam. That makes sense? Now, this will be the hard part. I don't know how good this is going to be, but what I do, as I think it's going to work. I'm going to go over here and we'll just mark this right here. This is our starting spot with just good old pure water. Is that all right? I'm going to put a mark there and just, if I can do it all with the water. See, I got it on the glass and I got it on the plastic and go, yep, that's my knockout spot. Is that all right? All right, so, and this is really fun working right over the light because I'm like, should have thought of that one before. Maybe like got rid of the rest of this. I'm just gonna get a bunch of sugar and put it in there. Gotta have the filter. That should work. All right. Pour away. I'll do table sugar. I want to put a boatload in there. Hopefully it'll go into solution now after all that mess. But we'll find out. So, if I get that right, and this truly twists light, then what I think I can do is once I get the light on and set the filter, it's going to be dark, but I'll twist it to get it darker now. Because the light's going through the first filter, twisting in the sugar. So to knock it out, I now have to get the twist. Follow? And that, by the way, this is the design of what they call the polarimeter. It's the instrument that can actually detect that this stuff is going on. So if you ever were, on break or something, you wanted to just, hey, show me that thing. I can take you up and show you an official polarimeter. Mine's kind of like I'm making it up, right? But we actually have them where they're detailed. They give degrees and all sorts of stuff, and they don't bind you like this one does. This is pretty rough. So let's see if we can get this into solution. See if we can twist some water. Be pretty cool. See? And the other piece of it is how concentrated this is. It twists it more. And then also is how long the column is. So I probably can't get it super concentrated in the time frame I'm doing this with you guys. So what I thought was I'd make a long column. So I get more twisting effect, right? And the thing I can't do is I don't have a D and L sugar. Those are a little more expensive to get them isolated. So this is sucrose, so it should be D, right? So I can do that. If I pop the L in though, it twists the other way. That's what this is saying, and that's true. So if D goes this way, when I go to put L in, I'll have to twist it that way to get it knocked out, which is really cool. And then there's a whole chemistry around this. You can imagine in the pharmaceutical industry, like these people that make these molecules, now all of a sudden they're thinking about handedness, and they've learned all sorts of tricks in the lab to like when I make it, it's going to come out with a certain chirality. Now you know that term. That means a handedness. And if they're doing carbohydrate chemistry, they want it to come out D if they think it's something that's going to be applied in the body, right? If it's a lipid, you want it to come out cis, but that's easier to see. That will have effects, which I talked about, right? Because we said cis stuff, does it have higher intermolecular force or lower?
Higher.
Does it act like it's branching or does it act like it's not branching? Cis is kind of the same effect as branching or not branching. Trans, remember? Yeah, cis is like branching. So what it does is it lowers the intermolecular force. So I can look at cis peanut butter and it's liquid, and I look at trans peanut butter and it's solid because it has a high boiling point. This thing I can tell boiling points are the same. That's just what made this problem so complicated when they first started looking at it. I'll kind of turn that towards you. Hopefully we won't both come out of here blind after today. That'd be awful. Now this class. Well, I hope Dr. Thompson. Okay. All right. So I got that. I'm going to put it right there. See how we do. It's pretty close to the same, isn't it? Did you see on here? Do you think that lines up pretty good? Sorry? Okay, so if this twists, then my theory is that when I start to twist the film, it'll actually go darker. Right? Because right now, in pure water, it would be the darkest it could possibly be. So here we go. No promises. We'll see what happens. You guys are all thinking I've tried this, and I swear I have not tried this. You think, well, surely you want to do this with us. And I'm like, yeah, I would. Game for anything. Or I'll see if I can get it dark enough noticeably. Ooh. I don't know. Anything? What's the consensus? Not too noticeable. Hey, will you turn off the lights over there? Maybe we'll try that and then we'll. Okay, that's definitely lighter, correct?
Yeah.
So you guys just tell me what you think the darkest is. Yeah. Man, this is not even close to what I thought this would be like. Okay, tell me when. Yeah. Is it?
Yeah.
Yeah.
I don't know.
Right there.
I'm not real happy with this demo. That was.
Is it because the light is diffused?
I think it could be the light diffused, and it could also be that I need to take out this piece. Like it's really throwing it off. So I'll play with that some more. Thanks for playing. Why did I pay you? Four others. Glad I didn't go and say, you don't need to buy those, I can make my own. Apparently not. Very cool. But yeah, good thought. Maybe it is the piece. So that was the idea. All right. So on we go. Now, this is the main take home. Can you identify a D sugar? Get away from the farthest away from the carbonyl group, right? The aldehyde or ketone. Still be chiral though. Don't go to that end carbon. On the right or on the left, right? In that Fisher projection. Correct? Mm-hmm. And then we know that D is the predominant form in nature. And then L it when it know what to do with it. Right? And that's kind of the end of this piece. And then also I'll have you just pick out chiral carbons, like out of any structure. And this might be interesting as you start to look at stuff. Now you look at and you'll see, like, even sometimes you can Google drugs and look at their chemical structure. You can look and you can see how many chiral parts are in there. And sometimes that's the key part. So, we're going to learn a few other things. We're going to do some monosaccharides. Okay, I'm going to just show you a few. I'm going to just show you a few monosaccharides and want you to know about them by name. So glucose is the one you should know. This is a single sugar, so I call it a monosaccharide. This is also called dextrose. So I want you to know that it gets that name thrown on it sometimes. They're interchangeable. This is your blood sugar. This is the thing that you need to get energy out of your cell. And I think that place is right, the mitochondria. Yeah, isn't that all your biology teachers like? Mitochondria is the powerhouse of the cell. Okay, good. It's needing glucose to turn it into energy. All right. So we'll kind of mess with that a little bit. Now, you will sometimes see monosaccharides where they turn into rings, and that's okay. So I want you to also see ring sugars and not get thrown off and go, hey, I still see a sugar in there. Because what are we looking for? What structural feature or functionalities in every carbohydrate? Lot of alcohol. So I look at this and I go, oh yeah, there we go. There's an alcohol, there's an alcohol, there's an alcohol, there's an alcohol. Oh yeah, I got an alcohol on almost every carbon. That's a carbohydrate. Do you know of all the biomolecules, this will be only for carbohydrates, so that's how you lock it down. And like when you're taking the test, if I show you pictures of things, if it doesn't have big hydrocarbon chains, 12 and 15, don't call it a lipid. Sometimes they're ringed, right? But you got lots of carbon, that makes it a lipid, right? Okay, cool. So I'm going to just show you, this could also be ringed up like this. Now, I'm not going to ask you to do D and all that on this, and at some point I'm probably going to come back and kind of reteach this a little later and show you a little bit about what they call the anomeric carbon that connects these rings. But for you, I just want you to see how I got from here to here, just so you understand it, that these are actually the same sugar. The body sometimes turns them into cyclic sugars. Or you might get them naturally in cyclic sugars and the body unravels them into straight chains. Okay? So here's the difference. In there, this had the carbonyl group, correct? And then in here I don't see a carbonyl group, but I do see a very unique functionality. Who can identify that? Pull your functional group table and tell me what that is. That's very much part of this test, right? I got it. I go to an oxygen, not a carbonyl, right? Just a single oxygen in the middle. I go to this side, I see a carbon. I go to this side, I see a carbon. What do we call those? Ethers. Yeah, these are ethers. So somehow that aldehyde turns into an ether. And here's how it works. So this is kind of wild. And it actually is, the actual mechanism doesn't, that's not the ether oxygen, it's this one. But what happens is that last chiral carbon there, it goes up there and it just basically attaches to that, and the H gets transferred up to the O. So that's a long process. You don't need to understand all that. But basically what's going to happen is when that reaches back and attacks that, you could see that that's carbon 5, that's carbon 5, O should reach up and connect to carbon 1. And then this thing, it took the H out there and popped it onto there. So see how it was yellow here? So yellow just so you can kind of see it. That's how those ring up. Now for you, all you need to know is, I see lots of alcohols, it's a sugar. I need to know that the aldehyde or ketone gets turned into an ether in these ringed sugars. All right? That's all you need to know. In the past, we've actually literally, and that's what this is from, we actually tried to get where students could learn how to do this, but this is super, it's kind of complicated. And we don't need, I mean, it's not that critical to what you guys are doing. Like, you're not gonna, hey, I know how to turn, right, a straight chain into a cyclic sugar. That's nothing you would do in the practice, so it's not that important for you guys. But anyway, so you can kind of look at these and go, okay, here's a glucose molecule. This is the glucose ring form. Same number of carbons, right? We're still 1, 2, 3, 4, 5, 6. These are 6 carbons, right? 1, 2, 3, 4, 5, 6. That's interesting. That's popped out there. But before I had a carbonyl group, now I have a ether. So that's that. So sometimes charts will put in this one. So here's your one monosaccharide I want you to be familiar with. This is glucose, also called dextrose. This is your blood sugar, right? Product of photosynthesis made by plants and stored as starch. We'll talk about that more. But you guys know this is our cellular energy. Here's another one. So this one's fructose. So this is interesting because this is made from a different kind of base. It's actually from a ketone base. So when the ketones ring up, they make 5 carbon sugar, or like a 5-membered ring. It's not 5 carbons in the ring, but it makes 5 members. But I just want you to know this. The word fructose is associated with sugars that are made from plants, and they're more natural. They're made from plants and fruits. And we're able to turn those also into glucose in our body. We have our own little factory in there that goes, hey, I can take glucose and turn it around and make it into glucose and use it if I need to. I'm sorry, fructose, I can turn it into glucose. And then here's the third one. This is the ribose. 5-membered ring. This is the stuff that makes the DNA strand. This is the stuff that makes the backbone for the DNA strand. So that's one of the 3 I want you to know, the monosaccharides. So I'm just showing you that when you look at the DNA strand, all along it is a ribose, phosphorus chain. That's what makes the 2 strands of the double strand. And we'll study this molecule in detail later, because it's your 4th biochemical group called the nucleic acids. All right? Good stuff. Now, I'm going to just, we'll get to this later, but the reason they call it deoxyribose is because we took one of these oxygens out of there and we knocked it out. That's the deoxyribose. We did it physically, not like chemicals, not like chemists did it. We did it biosynthetically. So that's where the deoxy comes from in there. One of the alcohols got knocked out and turned into just a carbonyl group, like a hydrocarbon group. Is this chiral anymore?
No.
Nope. Good. So, just real quick, and we'll get you a break, and then we'll kind of finish this. I just want to make sure you understand. I've got 3 monosaccharides I'm going to learn. There's glucose, used for cellular energy. There's fructose, can also be used for energy, but the source is from fruits and vegetables, right? Now they say it's found from honey, but if you think about that, that's because the bees hauled it over from flowers they were hanging out on, right? So, same idea. And then there's ribose, which is the backbone of the DNA, so that's the structural one, the monosaccharide. Cool. Take a break, we'll get back and we'll kind of finish up these carbohydrates. And then, so monosaccharides, we have 3 to 7 carbons. D and L anomers differ in the orientation of only OH group on the carbon farthest from the carbonyl group, right? D on the left, R on the right. D on the right, L on the left, right? And then also we showed you that they're cyclic forms. So something that's relevant to you guys instead of only just looking for like L sugars that you could taste, but that wouldn't totally be taken up in the body, they did a little chemistry on these glucose molecules. And what they were able to do is, what are these for? Does this sound familiar? Where do I apply hydrogen and platinum across things? What did we learn about that with lipids? What do they do? It's a catalyst, right? The platinum's a catalyst, and it could be either platinum, it could be anything out of this family, nickel, palladium, platinum. And you add hydrogen. And what does it do to a double bond? I heard it. It makes it a what? Single, because it adds hydrogen across the double bond, correct? Is everybody okay with that? So somebody used that same kind of application. They don't usually use platinum to be specifically, I think they use ruthenium usually, but same idea. They basically, what they're able to do is, if I added a hydrogen across this double bond, then imagine there's a hydrogen here, and then this pops up and makes an OH, that would add the H2 across the double bond. You see it? That makes a CH2 where it was a CH, and that makes an OH where it was previously an O, and it makes it into an alcohol. And these are sometimes just referred to as sugar alcohols. They're made in the lab or they're made in the industry, and they're the stuff that makes sorbitol. That's the sort of sugar-free gums that people chew, right? So that's, you with me? So when you start talking about sorbitol, now you know what you're talking about. So yeah, Trident, right? It's got that in there. Xylitol, sorbitol, you hear those sugars. They're sugar-free. It's the same idea. Because of the difference in structure where I don't actually have a carbonyl group, the part of my ribosomes that would be cranking that out and turning it into energy, they go, I don't know what to do with this, and just send it on by. But the taste buds, they still receive it as a sugar. And then the stuff in your mouth that breaks down sugars also doesn't work on it. So it's perfect. And so this is what you guys might recommend for people to chew if they, still want to chew gum and still want that sweet flavor. But that's what sorbitol and xylitol is. So it's a sugar alcohol.
If we can't use it, do we store it as bad stuff or get rid of it?
Yeah, so this particular one, it gets kicked out. And again, it doesn't really build up in the liver, it just gets excreted. Partially because it's very water soluble. All right, so these are called sugar alcohols, and I'd like for you to know this reaction because this is a reaction you already know. How do we do it? Add hydrogen across the double bond. We did the same thing with lipids, right? Cool. So, now how does your body do chemistry? So they take single monosaccharides. Yeah, see it? These are single monosaccharides. I remove, you see it in red, what do you think that's going to be that gets popped off of there?
Oxygen.
I got an OH, I got an H, what does that make? Water. You remove water and you make this glycosidic bond, which is how you start to bond sugar to sugar. Same type of reaction you've already learned, right? You've learned that hydrolysis, right? The removal of small molecule, right? So it's the same sort of thing. It's kind of like a hydrolysis reaction. So if I were to ask you, hey, gosh, what's the chemistry behind how two single monosaccharides start getting connected to make disaccharides and polysaccharides? It's just removal of water to make this glycosidic bond. All right? So the reverse of this, they also call hydrolysis, is the reaction that takes place during the digestion. So you can imagine that if you have a disaccharide, you're going to actually add water back onto there, right? And that's going to re-separate it out. But again, the glycosidic bond formation and the removal of water, they're both catalyzed. They don't happen naturally. I don't just take two monosaccharides and pour a little water on them and they become disaccharides. I have to have a catalyst to make that happen. Same thing about breaking it apart. So we learn with catalysts, what do you hear at the end of their name to know that they're actually a catalyst? What do you hear in the name?
Cat.
ACE.
ACE, right? So if I'm taking apart a sugar that's called sucrose, I would probably break it apart with sucrase. So just kind of keep that in mind. All right. So we're going to teach you just a couple disaccharides just so you're familiar. The first one is lactose. Is anybody familiar where that's found? Where does lactose come in?
Milk.
Milk. It's milk sugar, right? And some people are lactose intolerant because they don't have the enzyme that would break lactose apart. Why do you think that's called? Lactase. So they don't actually have a lactase enzyme. So when milk sugar comes in, they can't break it apart. So it doesn't really digest and kind of makes them feel, boy, that doesn't feel right in my stomach, right? So what's in a lactose molecule? Well, you got a glucose and what's called the galactose. But again, once you split it, then you have this food source in the milk sugar. That make sense? So of your disaccharides, I want you to know lactose comes from milk sugar. I want you to know sucrose because that's the common sugar like I was pouring back there, your table sugar. Important for you guys especially because you have a little bit of sucrase enzyme in your mouth, so it breaks apart right in your mouth and then the bacteria can jump on that glucose molecule and they can start to go woohoo, and they're chewing that up and spitting out something that's acidic byproduct. And that's why your mouth gets in an acidic condition. And that's what starts to tear up your, creates caries, right? Or cavities, right? All right. And here's the last one, maltose. So this is what's found in fermenting grains. So it's found in a lot of alcoholic beverages. Just thought we'd teach you a few so you just know some names and know what they are. Is that all right? That's all I'm going to ask of you at this point. I might come back and show you a little bit more about this alpha-beta linkage, but we'll save that till after the test. Like how those things lock across there, because it's going to be pertinent to how all this forms. So again, I'm just going to review just so everybody was on the same page and they understand. I now have monosaccharides, glucose, ribose, and fructose. Yes? And then I now have a set of disaccharides I should be familiar with. And basically it's where they're found, what they're used for, right? So out of this we have lactose found in milk sugar, sucrose found in common table sugar, and then maltose found in like beer and malt grains. Okay? And then we go to the next group, which is a polysaccharide. So, we already learned this idea that, look, if I have a repeating unit and I can just keep repeating and repeating, I make a polymer. So if I had a monosaccharide and I started polymerizing it from thousands upon thousands of those, that's called a polysaccharide. So again, I'm just going to teach you a few of those. Now look what we've done here. We kind of shorthanded this a lot because it looks like a disaccharide, but it's not. That's a polymer notation. They just kind of abbreviated. Should have been a parentheses, right, if it was what we'd shown you before. But basically the repeating unit is this. It also has repeating glucose in it, just over and over. That's what it's made of. And this is amylose or starch. And what we know to be starch is basically that's long-term energy storage because I can basically cleave that up and get all these glucose molecules out of it from my body. Okay, and starch comes from stuff like seeds, beans, potatoes, wheat and rice. Does that make sense? I start off with the simple glucose molecule and just polymerize it. And that makes starch. This one has an alpha 1,4 link. And again, I haven't really kind of discriminated what's the difference between alpha and beta, but we'll go back to that. And then here's glycogen. Really the same structure. It's got a glucose, it's got an alpha 1,4 link. It looks just like starch. The only thing is its source is different. That's it. So if I looked at glycogen or I looked at starch, it would look the same. The difference is starch is something we find, we grow in plants like potatoes. Glycogen is something we make ourselves internally. So if I have a lot of glucose building up, I might, through a pretty complicated pathway, turn it into lipids. So you can imagine that takes a lot because there's a lot of alcohols missing all of a sudden, right? So that's a whole nother mechanism. That's different. Or I might turn it into just a really quick, storage in my body of glycogen, which is just glucose all strung together. So I can just cut off that as I need it. That make sense? And that's usually stored in the liver and the muscle cells. So I actually have some glycogen in around my muscles or in my liver. It's just a place to tap in if I need some extra energy. It's a little faster to produce than if I go to a lipid, because a lipid has to have a whole biochemical pathway by which it turns back to a glucose molecule. Does that make sense? And then here's the famous cellulose. Again, repeating, glucose, the difference is it's actually what's called a beta 1,4 link. So the way that this kind of connects in here, which is really very, it's fascinating if you look at it, you look at the structure. The way that I easily identify it is these two ethers are oriented the same way in space. You see it? These are the same molecules, they're glucose all the time. I'll go back. Boop, boop, boop. Bear with me for a second. That got me out of the PowerPoint. There we go. Hold on. Okay. Here's starch. You see ether up, ether up. All right. Here's glycogen, ether up, the exact same molecule as starch. It just came from a different place, right? You guys understand starch is coming out from vegetables and plants. Glycogen I'm making myself. Now watch when we get to cellulose. Ether up, ether down, it got flipped. And that's what they call the beta 1,4 link. And this, my body can't do anything with, which is so wild with just that little flip. Then the body doesn't recognize it anymore, and then that becomes the plant cell wall that I can't digest. And so cellulose is the structural component that makes plant stalks, tree stalks, all that good stuff. Now, amylose accounts for about 20% of the starch, several hundred to 1,000 units, just so you kind of get a sense of like how big these starch chains are. And a lot of times this stuff tends to coil into a helix. I don't know if there's anything else that I find is that most, it's just interesting information, nothing that I necessarily kind of quiz you on, but I'll just leave it at that. All right, so there's kind of your carbohydrates. Any questions? Yes, no? So when you revisit that, what you're going to really work on is kind of, first off, learn the monosaccharides I want you to know. Learn the disaccharides I want you to know. Learn the polysaccharides I want you to know. So those are pretty straightforward. And it's mostly about, here's the name and where they're found. Right? Make sense? And then beyond that, I did show you a little bit about the stereochemistry, about the DNL and the predominant form in nature. So not a ton about that. And also just if you saw the molecule, you would go, that's a carbohydrate. Right?